Hello, my name is Mona, and today I will discuss how we develop a method to enable vessel density mapping of brain small vessels using 3D high-resolution black blood MRI at 3T. I do not have any financial relationships to report. In this study, we are interested in developing a framework for localized 3D vessel density mapping of small vessels of the brain from 3D high-resolution black blood MRI images acquired at 3 Tesla. Recently, high-resolution 3D black blood MRI with sub-millimeter spatial resolution has been proposed to visualize lenticulostriate arteries or LSAs. Utilizing this optimized MRI sequence, 3D segmentation and quantification of LSAs is feasible. In this study, to enable 3D and localized comparisons of brain small vasculature across populations, we developed and evaluated a novel framework by means of automated vessel segmentation and nonlinear registration. For data collection, paired black blood MRI and structural images were collected from 16 participants and divided into younger and older groups ranging in age from 22 to 33 and 61 to 81 respectively. Image volumes were acquired on a Siemens 3T Prisma using T1 space sequence. For input into the vessel segmentation model, the raw images underwent several pre-processing steps. Initially, black blood MRI and MP-RAGE images were skull stripped. Afterwards, black blood MRI were pre-processed by bias correction and denoise via non-local means. Vessel enhancement was performed by multi-scale Hessian-based method, and then a binary vessel mask was computed by thresholding the vesselness map. After that, a vessel density image or VDI was computed for each subject by convolution between BVM and a 3D average kernel. To pull the VDIs from all subjects into the MNI atlas, we implemented a two-step registration approach. Initially, we robustly co-registered black blood MRI and MP-RAGE image pairs using 3D affine registration with 12 landmark points. Using this transformation, VDI was first reversely transformed to MP-RAGE space and then nonlinearly transformed to the MNI atlas using a B-spline transformation. After nonlinear registration, all VDIs were normalized and a p-value map was generated by comparing the registered VDIs of the two study groups at voxel level. The performance of three multi-scale Hessian-based methods have been compared on a synthetic image. It can be clearly seen that compared to Frangi and Sato's methods, Jerman filter is more robust to noise and also has higher vesselness measure at response profile and at vessel boundaries for both small and large vessels. In this slide, we have examples of the three Hessian-based methods on image patches from high-resolution 3D black blood MRI. The visual assessment of the three methods showed that it is feasible to perform automatic segmentation of the small vessels in regions with varying contrast. Jerman especially performed better compared to the other methods for low-contrast vessels of a small size. Here we have samples of segmentation results of 3D brain vasculature of the winning method Jerman on the subjects from two age groups. Vessel density differences among individuals from the two groups is visible by qualitative comparison, where age group has a sparse vessel network compared to the younger group. For quantitative evaluation, we have the box plots of normalized vessel density by the head size, where we can clearly see the significant difference of vessel density measures between the two age groups. Additionally, voxel level statistics was performed to demonstrate the localized significant regions with decreased vessel density in the age group, which are highlighted by red-colored clusters. The results of automated vessel segmentation with Jerman filter and nonlinear registration seems very promising for localized detection and quantification of cerebral small vessel density changes, though the results should still be observed with caution because further parameter optimization and testing on a larger cohort is required. Additionally, the current field of view is limited to partial brain coverage due to relatively long scan time to achieve high resolution. In future, we will incorporate more advanced acceleration techniques to enable increased coverage in clinically appropriate scan times. Finally, I would like to acknowledge the many people who made this work possible. Thank you to the volunteer participants, the Loft group at Loni USC, and our collaborators. Lastly, this work was supported by grants from NIH. Thank you all for your attention.